Welcome everyone, and thank you for joining us for the 38th Annual Succulent Plant Symposium. My name is Sean Lawmeyer, and I'm the Collections and Conservation Manager here at the Huntington Library, Art Museum, and Botanical Gardens. For those that are new to this event, this is an annual series of talks that we host to discuss all things related to cacti and succulents, including horticulture, design, and botanical research. Over the years, the symposium has featured some of the most notable plant people of our time, and has served as an informal educational lecture series for the desert collections here at the Huntington. This is now the second year we are offering a remote version of the symposium, and this year we have decided to offer a short program focused on growing succulent plants, with the specific aim of highlighting plants that are difficult to propagate. Today we are joined by 6 seasoned growers from across California to discuss some of the plants that have been most challenging for them to grow. Please feel free to ask questions during the Q&A feature, and we will do our best to answer these questions either after the individual presentations or during the group Q&A at the end. Before we get into the program presentations, we wanted to give you an update with regards to the Upper Desert Garden construction project that many of you may be aware of. To tell you more about that, I will turn it over to Seth Baker, our garden's principal designer.
Good afternoon. I'm again Seth Baker, and I'm garden's principal designer here at the Huntington. And I spoke last year about our desert garden entry project and a little more in depth, but just wanted to give some updates today as far as things that have transpired and things that are being planned. So let's jump in. So again, desert garden entry project, and just kind of a look at our goals is first and foremost to really make an entrance because of the historical nature of the property. There was really never a formalized entrance, and in part to ease kind of wayfinding in a more intuitive way. Really want to announce this is the desert garden, and so we aim to do that. And then one of the other big goals is to expand access, and so that's both to open more areas of the garden to showcase more of our very vast collections, but it's also to follow universal design principles. And so that's really about designing experiences that are enjoyable for everyone. So thinking about both strollers and wheelchairs, walkers, making really convenient and beautiful experiences for everyone. But also to make improvements in the garden, and so really to create a more immersive experience, and then create opportunities for learning and interpretation, improve horticultural conditions, and rehabilitate the conservatory and install new infrastructure. The conservatory we really see as being an interpretive hub for this magnificent garden. And ultimately all of this is to cultivate a renaissance in the desert garden. So let's jump in. So this is the rendering I showed last year of really trying to make a punch, that statement of this is the desert garden. And one of the objectives is obviously to cram in a lot of diversity and show it off, but also do it in a way that has some cohesion. So within the plant palette that John and I have been working on, it's really finding things that have some commonalities. So rosette forms or very linear things like the palms or aloadias, and also just a color palette that maybe we'll pull it all together. Okay, and so then diving in to the plan here, just a refresh of this. You have the Celebration Garden fountain at the lower right of your screen, and that will act as the formal entrance. And so we're taking visitors as a part of that immersive experience off. Currently you go down to the road and then just kind of drift into the desert garden. This immediately pulls us off to the left, and then down to the conservatory where we'll be adding a epiphyte shade house where we'll expose much more of that collection that's largely been behind the scenes, and really work out as I said, the Desert Conservatory as an interpretive hub. And it just the footbridge and small gathering kind of micro amphitheater area, and ultimately then connected in with some of our more accessible path systems around the property. To kind of review what we're doing to the conservatory, this is more a diagrammatic look because we've shifted some things around since this was made, but this largely explains what we're going to be doing and stripping it down to its most essential structural elements, making some new fenestrations, some insect screening, and then retractable sides for kind of diurnal control, and then a new acrylic sheeting on the outside, and then surrounding it with a walkway system that allows people to view in from the outside, and then a shade structure that will cover the patio area there on the right, and then we'll also be adding the epiphyte shade house. And so here is kind of a look at some of what we've been up to in the last year or so. We brought in tons of new garden soil. And this is a crew that James Brumder helped arrange, bringing in all this glorious new soil. Here's John Traeger surveying some of our rocks that we've harvested from elsewhere in the garden and have had behind the scenes. Here's Shoddy and some of his crew working on rockery construction. And so those are really starting to take shape now. Of course, Tom Carruth surveying our progress. And then just kind of a look at the plant palette that we're anticipating. Again, I was talking about those repeating forms and kind of a cohesive color palette. So those codices, also those very vertical elements and rosettes that will be those larger repeating forms to bring continuity to the look and feel. So the aloe went in, a little over a year ago. We've started recently, again, doing more of the rockery. Then we've craned into place some palms both at the north end of the conservatory. So those persistent leaf bases on the palms will act as substrates to grow up some of our epiphytic cacti collection. Of course, the shade structure will be going in as well. We also craned into place a series of 5 palms closer to our Oxford entrance, which will serve as a backdrop in the more eastern areas of the garden. But they also are adding a great deal of depth. So these are some Brechia armata hybrids. And this is just a quick walkthrough of some of the progress, some of the rockery work, this new gentle path taking us down into the garden. And hopefully you can get a sense of how immersive it is. We're cutting out the views into the palm garden. So it's fairly limited. There's those new palms, being the epiphyte house. And then this really spectacular specimen craned into place at the same time that we were doing the palms. So Aloe Dichotoma from one of our longest standing volunteers in the gardens here. Very many of you know, well, she needs no introduction for so many of you, Sandy Chase. This Aloe Dichotoma has been growing for decades in her front yard out in Selmar. And so she donated it along with Artie Chavez to us. And it's certainly the best specimen I've seen, and people that know a whole lot more than I do all seem to agree. So here it is getting craned out of her front yard and really great sandy soil out at her house, but also made it move around out in the box a bit and getting prepared for transport once it arrived here at the Huntington. And then the next day, pulling out the big crane. Here we have a bit of a video of it coming up and over the conservatory. And so this is going to create a beautiful anchor for this new patio at the east end of the conservatory, which provides just great prospect over the garden. And this is just a great bookend there. That said, this was Sandy and Artie's donation, but getting it here was made possible through a donation of Marge Richards' estate. And there have been so many people that have stepped up and helped really get us to where we need to be with this project. Bill Wilk among them. I know so many of you are familiar, and yeah, I encourage you to help in the ways that you can. And here you see it getting covered the first day, just protecting it wasn't compassed properly. And then John and I spent a good hour and a half getting it finally positioned. Here it is currently, but we'll be out with braces here soon, and then kind of sculpting a landscape around it once we have it braced and remove the box. Again, I hope that a lot of you can see the promise of this project, and yeah, just keep following along, and thank you so much.
Our first speaker today is Ernesto Sandoval, who is the curator and manager at the UC Davis Botanical Conservatory. He describes himself as a Jose of all plants, master of none, and although he doesn't consider himself to be an expert in the genus Dorstinia, he'll show us what he's seen and discovered over the years for the successful propagation of Dorstinia gigas, which is the subject of his talk. He received his bachelor's in botany from UC Davis in 1996, but learned horticulture practices from the age of about 14 by assisting with his father's mow, blow, and go style gardening business in the San Fernando Valley. He's made it his life goal to understand the biology of plants as it relates to their horticulture as well as their enormous diversity. Welcome, Ernesto Sandoval.
We've distributed some of the Dorstinia gigas in several ways. In this case, this was a batch that I donated to or exchanged with the Huntington for their ISI, including Dendrosicyos in the background, and a few other growers as well that I distributed to back when I had hundreds of seed a number of years ago. One of the seedlings, some of the plants went to rare succulents in Rainbow, Petra Christ's rare succulents, and she planted them in some big containers, which I don't have big containers to grow mine in, so they're a lot smaller. But this is a massive individual that she has that was one of our seedlings, and it's actually quite a seed producer. So her and I have exchanged some seed in the recent past. Okay, so propagation from seed, okay, or propagation, we're going to start off with seed. So it turns out that this plant is actually we treat it like a tropical plant. And I have this talk is available. I think Sean may have posted the link, or I send him the link. He'll be posting the link to my lecture and other lectures. And I have a more extensive lecture on seed propagation of succulents, but right now I'm focusing on Dorstinia and it being a tropical. And you know, you take the seed and plant them any time of year, okay? Because it's a tropical or a subtropical plant, you want to keep the soil temperature warm, so 70 to 80 degrees Fahrenheit. So in a heated greenhouse, heat mat, or under grow lights, okay? And you know, again, it's tropical or subtropical plant. And other plants that you would treat like this, you know, tomatoes, peppers, basil, and then succulents, aloes, welwitschia, stapeliads, tropical cacti, okay? So I'm going to talk about cuttings at the end of my talk in the last few minutes that I have, making cuttings of Dorstinia, but you treat it as a normal plant, as a leafy plant, where you do it in a humid environment and use a mist bench or aeroponic propagator that I'll talk about, okay? One of the things that's really important when you're doing seed propagation is to have your seed under bright but diffused and indirect light. So you'll have them under shade where it's warm if you're doing them outdoors. if you're in Southern California and you're doing it in the summertime or something like that, and the temperatures are right. But you never want to have direct light on your seedlings. It's much better to have bright diffused light. Okay. Fluorescent lights or LED lights are a great way to do it. Some really good LED lights these days. Okay. I like to use some temperature monitoring equipment. So there's this thing called the Kestrel that I use to hang it near my plants where I'm propagating, so I can do some logging and tracking of temperatures, etc. Okay. One of the things that's really important when you're doing seed propagation is having a good, well-draining, fast-draining soil mix. So something called a succulent mix. And I'm going to talk about the sand here as I go along. This is a sand that I use to mulch my seeds with. Okay, so I'll show you that. Here's the UC Davis mix. It's 3 parts 3/16 pumice. We were using lava rock. Actually, what you see in this photo here is lava rock, but we switched back to pumice. It's much better than lava rock. And the mesh, the number 12 mesh sand, a little bit peat moss, and a little bit of redwood compost for nutrient holding. Okay, but basically it's a very well draining mix, and it's really good for seedlings and normal plants. Okay, one of the things that we do, especially when we're propagating from seed, is we pasteurize the mix. So we have it for at least 30 minutes at 180 degrees Fahrenheit in a big autoclave. But at home, what you can do is you can take your soil and your moist soil and put it in the microwave for about 5 to 10 minutes, depending on the strength of your microwave. And that kind of pasteurizes, it doesn't sterilize it, but it kind of kills a lot of pathogens off. And it's much better when you're doing, especially succulents, especially the drier succulents. You want a good, clean, semi-sterile, really what's called pasteurized mix, so that you don't get a lot of pathogens attacking your little baby plants. Okay. So here's how we germinate the seed. We do it in Ziploc bags, or when I'm doing a lot of seed, I'll do it in a big tub, acrylic tub. But I call this a Ziploc a baggy technique. And here's some young plants and some mature plants there out of the bag. And here's a little trial that I did with the Derstenia, where I put 50 seed in a bag, and I'll show you how exactly I planted them in a second. And then 50 seed out of the bag. And the one out of the bag, I had to water it like every day. And you can see how much better germination I had because I had constant moisture in a humid environment, in like the bag there. And so I can't emphasize this enough when you're propagating seed, is to have really good constant moisture. And by having it enclosed means you don't have to water it every day. And you can see over here, I was doing this in a low humidity environment. And so I really had to water every day to keep it moist, and it really was not enough in the foreground. But the one in the bag, I'd never had to water that. So, and what I'll do is that when I germinate the seeds, is then I'll crack the bag open by taking the top and sort of flipping it inside out and letting a little bit of moisture escape over a week or two, so that it sort of like drops in humidity to ambient humidity, and then I can pull my little seedlings out of the bag. Okay. And there are some that are still in the bag. You can see, droplets, and it's very humid in there. But when they're at this stage, I could then crack the bag and start dropping the humidity. Or if I'm doing it in a grow tub, cracking the lid so that the humidity starts dropping in there over the course of about a week or so is really good. You don't want to just rip them out of there into dry air and have them get sort of burned from the rapid change in humidity. So I want to play this video real quick and show you how I plant seed, because I think especially for beginners, this is really helpful. So I'm taking my nice pasteurized mix there, and I'm putting him in a pot. So potting it up, dropping it in the pot, and then I'm going to level it out. Okay. So I can drop my seed. And in this case, I'm actually doing Dendrosychios, which is a cohabitant of Derstenia gigas on the island of Socotra. But I take my seed and I sprinkle them around, try to get them, a little far apart. When I do a 3-inch pot, I'll do 25 to 50 seed of something, depending on how big the seed are. These are Dendrosychios seed. They're about 3 or 4 times as big as Derstenia. So I only do about 25 seed in there, but I could do like 50 seed of Derstenia. Now I'm mulching them with sand, And what's great about using the sand is that it lets light through. So I can put it on pretty thick. I can do a 1/4 inch, 1/8 of an inch, and level it out. And now my seed are going to be trapped in that layer between the soil and the sand. And they're going to get lots of light, but they're going to be trapped down there again to keep it humid. And now I'm going to water it from above with a nice watering can, fill it up, drain it through. Some people like to water their seed pots from the bottom so that it just wicks the water up and then you don't hydraulically mine the soil. Labels are always important. And now what I'm going to do is I'm going to take it and I'm going to drop it inside of a bag so that I don't have to water it again. Okay. And if I do this in the right temperature, I will have good even germination where like most of the seed will germinate like in just this nice little wave, like grass seed or whatever. Okay. So there we are. And that's how I do seed like 95% of the time. Okay. All right. Let's continue. And I gotta skip through this. There we go. Get to the next slide. Okay, so soil, then seed on top, sand as the mulch, then water and bag for constant moisture. Okay. All right. Here's the slides about I was doing some pollination experiments, trying to figure out if I could get some of my F1 seedlings to backcross, etc. The data is inconclusive. Okay. And these guys spit the seeds out. So I kind of put these little mesh screens over the plants to catch the seeds. And really, some plants are functionally female, other ones are functionally male. They have both parts, but I don't know what's going on with the genetics of hybridizing these things, okay? But now I just want to finish up talking about cutting propagation, because I know I'm limited on time, right, Sean?
If we can just wrap it up, that'd be nice.
Yeah, great. So when I do cuttings, I treat them as a normal plant. And I'm going to show you how I do it. Here I'm making some cuttings using rooting powder. So I used Hormex 3 and Hormex 8. Make my cuttings. I actually put them in vermiculite perlite. Put them on a mist bench. All right, that's one way that we propagate them. Here's the cuttings kind of moving along. Okay, and then another way that we do it is Aeroponic Propagator. And what you can see here is here's some Derstenia gigas cuttings that I did in this Aeroponic Propagator. This unit has water in there, it has microjet sprinklers that suck the water with a pump and spray it on the cuttings that are dangling above the water and getting sprayed with water. So it's really air with a splash of water. And just to show you that these things like water and lots of oxygen, and they root really well this way. Thank you very much.
Thank you, Ernesto. A couple questions in the chat. One, do you ever see monstrous growth form with Derstenia gigas?
Not with Derstenia gigas, but I'm hopeful that one day I'll get a seedling that does that. Yeah.
Another one came up, it said, why is, I think you said, why is pumice preferable to lava rock? Did you say that?
Yeah, so it turns out pumice, holds about 15 to 20% more moisture. And as it breaks down, it really is mostly what is it, calcium and some other relatively inert materials. Whereas pumice, for some reason, a lot of growers have found when they keep things kind of dry, that the lava rock, the lava tends to be toxic to plants. It slows down their growth. So pumice seems to me a much better coarse material to use in a mix than lava. Lava works, but pumice works better.
Thanks for clarifying. We can come back towards the end with if you have other questions for Ernesto's presentation. Moving on, we'll move on to our second presenter today. Our next speaker is Joe Stead, who is a full-time horticulture coordinator and a part-time instructor at Orange Coast College Horticulture Department in Costa Mesa, California. He's worked there since 1994. He has a passion and expertise. His passion and expertise are in the propagation and hybridization of many types of plants. He has co-authored articles published in the CSSA Journal, and his succulent design was featured in Sunset Magazine. He teaches an 8-week course on cactus and succulents and their use in the landscape. And he's currently working on breeding many new hybrids of Boswellia. Welcome, Joe Stead.
Thank you, Sean. Can you see me? Can you guys hear me? Hello.
Yes.
Okay, great. All right. My talk is on Burseraceae, and my title is Propagating Against All Odds. There are over 100 species of Bursera, and they come from the desert Southwest in the United States to Mexico, Central and South America, and to the Caribbean islands. There's a picture of me in front, or besides Bursera multijuga, and this is kind of what I want to talk about today. Those of you that have been to the Inner City Show at the LA Arboretum, you've probably seen this plant. This is Bursera multijuga. It was a bonsai plant by Michael Buckner. And every year when Michael would enter this plant in the show, of course, it would win lots and lots of accolades and ribbons. But after the show, Michael would come over to me and say, I want you to do cuttings of this. And I was thrilled to, but every year I would take the cuttings, take them back to the horticulture department at Orange Coast College. We have a really nice propagation house, mist and fog, and tried to root these probably 4 or 5 years in a row and was never successful. So it was very frustrating, but just in the last few years, I have a very good friend. His name is Jason Islamier, and he had some of these plants. He gave me a small seedling, and I've been growing that for many years. And mine was up to a big enough size that I could take some cuttings. And he had a couple plants in the ground. And so he gave me some cuttings also. So here they are, and different sizes. This is in at my hort department. So here I've got my dip and grow hormone at a 1:10 ratio. I found out that these larger cuttings are always the best ones to use. And I take a very clean razor blade. I make a 15-degree cut just below a node. And then I dip them in the rooting hormone for 3 to 5 seconds. Okay. So here's what I put them in. This is my particular mix. Like I said, I've got a really nice propagation greenhouse, so it makes it so much easier. In the bottom, these are 4 1/4 inch pots. In the bottom, I put my propagation mix, which is basically a potting mix and mixed with perlite. And then that's about 1/4 inch in the bottom. And then I add pumice and perlite mixture together. You can see in the top up here where the cursor is, and I've dibbled holes in there. And so what I do is I make sure that all this pumice and perlite and everything is nice and moist. Make the dibble, and then I insert the cuttings. What's really important is when you put these cuttings in is make sure that you firm that pumice around the cutting itself, and so it doesn't move. You do not want them to move at all when they're trying to root. It's very important. So this is one of the cuttings that I got to root, and this was just this last month. And it's interesting that this cutting is fairly big around, and it's nice and tall, and that was the secret to this particular one. When I got those cuttings from Michael at the show, they were all very, very small, thin, and they were the new growth of that year. So you need actually second-year growth, or even further on, maybe third-year growth, to get these things to root. So so I have a propagation greenhouse. So not everybody has this. So just a while back, I went down to my friend Jason's, and Ed Reed from Cal State Fullerton came along, great propagator himself, came along. And so we did some cuttings. And what we did was, here's another bursera. This is Linoa. And the best thing that I found about a lot of these burseras is it's best to try to leaf them or to root them out when they are not in leaf. And so this particular plant in the garden was not in leaf. So we took cuttings, did the 15-degree cut. Here we used a Clonex rooting gel instead of the dip and grow. And here we go. Now this is a different method, and I know that Jason has rooted thousands of bursera cuttings in the past, and so I really wanted to see this method. So we're sharing this with you today. So we took a side cut of this 4-inch pot, and what he does is you put some dry perlite in the bottom. The next layer is 50% potting soil mixed with 50% perlite. You can use, say, like a Miracle-Gro potting mix, or a one that he uses, I think, out of Home Depot, or it's called Vigoro. And these are really good mixes. And then you put a layer of perlite, another layer with pumice, and then the potting soil again, and then the pumice up at the top. So if we look at this picture here where the cursor is, you can see that there's already the perlite at the bottom, the pumice and the perlite mix is below that. Then you add the perlite as a layer on top. You dip the cutting, then you set the cutting there, then you add the pumice, okay? And that will, you firm that down a little bit. Then you add, over at this slide here, you add the 50% potting soil and the perlite on top of that. And then at the end, you add pumice. And then what you would do, okay, so here's another one. We're gonna show you this one. This one is Bursera kerberi, which is another really nice bursera. And this was in full leaf. So you know, here we have an instance where, okay, I want to do some cuttings, but this plant's in full leaf. So we're going to take some nice size cuttings. What we're going to do is we're going to remove most of those leaves. We're going to leave a few small ones on there, but we're going to remove most of them. We're going to cut it right below the node at a 15-degree angle here, as I'm pointing. And then there's a branch here. I've kind of taken it back a little bit. And it's nice because that's going to actually help to stabilize the plant in the pot. It won't move around at all with that slight little branch there. Okay. So this is a larger cutting. So we're using a 1-gallon pot, and we put a little bit of perlite in the bottom. Then we've put up about halfway up the pot the potting mix and the perlite. And now we've stuck the cutting we've dipped the cutting in the rooting hormone. We add the small perlite there, okay, as a base. And then we pumice it around just to hold it a little bit, all right? And then we add the potting soil almost up to the top. And then we top dress with the pumice. And that holds it really secure. The plant will not move very much, okay? You usually wait until the next morning to actually water this in. And then you would water every other day. And the secret with this is do not let the soil dry out. The way that this is being done by Jason is he can leave these plants outside, okay? Getting full sun. I would do it in a shade house myself, but he's done this, and these can be out in full sun. They're not gonna, they will do just fine. Okay. So another way that we can root some of the burseras is from root tubers. Okay. These tubers can be removed from the plant. As you can see in this picture here, they look like little potatoes. You can pot up these tubers in your regular succulent mix. I have like a 50-50 succulent mix, and you want to leave at least 1 inch of tuber above the soil. You can, some of these you could just cut, they will come right off the root. So there's very little wound there. If there is a bigger wound, then you can seal that with the rooting hormone. The rooting hormone gel works good for me. And then you want to move this into a very moist environment with nice warm temperature and high humidity. And in about a month or two, you will see shoots emerge from the tubers. And after the shoots gain some size, then you can start to fertilize. So the next slide will show you. So this is in my propagation house. This is on bottom heat. And you can see that on the left, you can see these little root tubers. And some of them don't have too big of a wound. This one down here has a little bit of a wound. Sometimes they will actually shoot right underneath the soil. They won't even come right out the tuber. They'll come out the bottom of the tuber. This larger one up here, this is Bursera morolensis, and this was rooted last year. But these cuts are getting big. So 2 months later on the right, you can see that all these smaller ones are rooting up nice. It's a great way to get some new cuttings. Okay, seed. I was just going to talk about seed real quick. I was at Ed Reed's place in Cal State Fullerton and had a few seeds on Bursera vasquez-yoncii, which is a wonderful species of Bursera. I soaked these in water 24 hours. I sowed them November 29th, 2020, and they germinated like 10 days later. This is what the plant looks like in April of this year. It's much bigger now. It's already got a second branch on it. Okay, here's a fun one. The seeds, a lot of times you can soak them. I soak these in water for like over a week, and you can see the radicals are coming right out in the actual water, which was pretty funny. So I was like, oh, great. So I sowed these up. Almost all of them germinated, of course, within a few days. This is an unknown species. Jason didn't know what it was, but these are nice little plants, and I'm excited to grow these on. Another way to propagate these plants is air layer. If you look at on the left, you see this little red diamond here. This is where I did an air layer, and I'm not going to go into the details of air layering, but because we don't have enough time. But I air layered this little section here in April of 2019. And if you look on the right, if you looked at this cutting here and just kind of turned it sideways, you can see that this branch, this branch, this branch are these 3, 4, 5 branches up here. So just turned it, and here it is, just a few months later. So another great way. Okay, that's it. Special thanks. I want to give special thanks to Jason Islamieh and Ed Reed for sharing friendship, knowledge, and plant material. Also to Orange Coast College Horticulture Department, who's let me have fun. This is a picture of Jason Islamieh's book. It's one of the only books that I know on Bursera, and it's a cultivation book, so it's a good one. Anyway, thank you.
Thank you, Joe. Fantastic. We have a couple questions. Let's try to get one. Are there different types of rooting hormones? And if so, which are preferable? Are some more effective than others?
Okay. Yes, my preference was dip and grow, and it's been that way. I used to use the rooting, the powders, but they seem to lose their effectiveness after time if you don't keep them stored properly. The gels work really well also, the rooting gels. That Clone X works fine. I like the dip and grow because it has IBA and NAA rooting hormones in it.
Thanks, Joe.
Okay.
One more,
Yeah.
We can do others after. You say a larger cutting, how long are they in? How long are you talking about in inches or in diameter? When you say
You could, I would say 1/4 inch to up to 4 inches in diameter. You can root. I did a plant from Tim Harvey a couple years ago. It was a pretty good. It was at least 1 inch in diameter. I put it in pure perlite / pumice. Didn't even remove any leaves. stuck it in a 1-gallon pot, put it in the propagation house, was not under mist or anything. And by the end of the summer, it was fully rooted. So.
Wow. Okay. Fantastic. Thank you again, Joe.
You're welcome.
We
Thank
will
you.
move on. Our next speaker is a board member of the Cactus and Succulent Society of America and has been curator at the Ruth Bancroft Garden for the last 41 years. He is vice president of the San Francisco Succulent and Cactus Society and vice president of the Institute for Aloe Studies. He has authored numerous articles on succulents and cacti with over 300 published, and he has contributed many photos to dozens of books on succulents. Please welcome Brian Kimball.
Well, hello, everybody. In 1972, a small echeveria was collected by Charles Glass in southern Nuevo León in northeastern Mexico. It was subsequently collected by others, including Dave Grigsby, Kitty Sabo, Alfred Lau, but was not named until Myron Kimnack published it as Echeveria cuspidata variety Zaragoza in the Cactus and Succulent Journal in 2005. And the issue was volume 77, number 1. The typical variety, which is now designated as cuspidata variety cuspidata, is an older name published by Joseph Nelson Rose in 1903 from a plant collected by Edward Palmer in 1902, and that flowered in Washington, D.C. in February 1903, enabling the description. It came from near Saltillo in southeastern Coahuila, and it also occurs a little bit farther west near Parras, Coahuila, and was redescribed as Echeveria paracensis by Eric Walther in 1959 and appears under that name in his monograph on the genus. But it's now considered a synonym of Echeveria cuspidata. In this photo by Myron Kimnack, we see 3 plants of Echeveria cuspidata Zaragoza on the top row and 3 plants of Echeveria cuspidata cuspidata on the bottom row, so you can compare the two. You can see that the leaves on cuspidata variety Zaragoza are shorter and more truncated and puffier, not as flattened, and with distinctly darkened tips. Like Echeveria cuspidata cuspidata, the variety Zaragoza was named twice, since Kimnack gave the name Echeveria cuspidata variety Gemula to a second plant that proved to be a synonym. Here we see a plant in habitat near Zaragoza, Nuevo León. And actually more formally, the town is known as General Ignacio Zaragoza, distinguishing it from other towns called Zaragoza, such as Villa Zaragoza and San Luis Potosí. Here are more plants in habitat, and usually they grow as a single rosette, like the plant on the left, but sometimes they become multi-headed, and you see a double-headed one there on the right. They grow exclusively on gypsum, which is calcium sulfate, while cuspidata variety cuspidata grows on limestone, calcium carbonate. And the plants oftentimes are found growing in a vertical base, such as you see here. The rosettes are small, usually less than 4 inches, and oftentimes more like half of that. And they can have really quite a few leaves in the rosette, and the rosettes are very neat and attractive, as you can see. And here is a plant growing in a very shaded position. And so the rosette is looser, more open than it is in plants that are more exposed. And you might suppose this was a young plant, not that many leaves in the rosette. The rosette's not that big. But if you look on the upper left of the screen, you can see its stem, which descends down. And obviously, it's been growing for a very long time. And so they can live to be quite a few years old. And here are photos of Echeveria cuspidata variety Zaragoza in cultivation. And you can see what it looks like in flower. So the flowers are shorter than those of cuspidata variety cuspidata, but the flowers are really quite similar. More of a pinkish red on the outside and quite orange on the inside. in less light, the inside is more yellowish, yellow-orange, rather than the deeper orange that it is in more sun. And for a close-up of the flowers, there you see the flowers. So this is a very beautiful plant and well worth cultivating, but it has not become common in cultivation. And I think perhaps part of the reason for that is people have difficulty growing it. I know I did when I first started growing it, and found that the plants would sort of sulk and not want to grow very much. And then I had the idea, it grows on gypsum, why not apply gypsum to the pot? So I got powdered gypsum and sprinkled that on the pots, and almost immediately the growth improved. And so that is a secret to growing this plant, but also might be applied to many other plants that are gypsum endemics, such as Echeveria hituensis, or many of the other things like Geohintonia and Astecium hintoni that also grow on gypsum. So well worth trying. Well, I hope you enjoyed seeing these pictures, and I have sent seed of this plant to the Huntington, and I hope perhaps it will occur on a future ISI list so that everybody can grow it.
Thank you, Brian. Appreciate it. Our next speaker has been growing cacti and succulents for 43 years. Has made a point of being active in the local and national cactus and succulent societies since the 1970s. He's currently a member of the Central Coast Cactus and Succulent Club, for which he was a founding member. Along with his interest in cacti and succulents, he enjoys photography and travel, and these interests have come together in his many botanical explorations of the Western US, Mexico, South America, Africa, Madagascar, and the Middle East. Many of his photographs have been published as illustrations for articles and covers of the CSSA Journal and Hazeltonia, as well as the Timber Press book on succulents of the world. He has also recently published articles on Pediocactus and Areocarpus in the Cactus and Succulent Journal. Welcome, Rob Skillen.
All right, thank you, Sean. So I hope that what you're seeing now is the title slide to my presentation.
Yes.
Okay, great. So I am talking about propagating Adenia, and that is Adenia, not Adenium, two very different genera. I'm going to talk a little bit about both the success and frustration that I've had in doing this. So I would first like to talk about propagating Adenia from seed. And the important issue here is that most of these species are dioecious, so you're going to need to have a male and a female plant. And it's very easy to just pluck a male flower like we see on the left and daub the pollen onto the female flower that you see on the right. And very soon you'll see the fruits developing. And so that is a very easy process. Once the fruits are ripe, you can harvest the seed, and you'll find that the seeds are encased in a translucent membrane and that there's some juicy pulp around it that's either clear or in some cases red. And you need to clean that pulp off. And it's often recommended that you also clean off that membrane. That's a difficult task to accomplish, and many people who are successful growing from seed don't do it. So you might be able to get away without it. It's also commonly recommended that you must age the seed for about a year before it will germinate. But that's not necessarily required either. In fact, I've got that little inset picture in the upper right, which is a flat of Adenia glauca. I produced the seed several months ago in the spring of this year. And this is a photo from a nursery that's growing the seeds from just a few days ago. And they have neither aged the seed, and I don't believe that they put any effort into trying to get that membrane off either. So you can see that you can be very successful without either one of those tricks. What you do need to do is make sure that you avoid hot conditions after you've sown the seed. Don't use a heat mat. Place it in the cool portion of the greenhouse and cover it with some sort of a shade cloth or some manner. Keep the direct sun off of it. It must be kept continuously damp. If it's in a seed flat like we see here, missing it every day is fine. Don't let it dry out. And then germination will be erratic. They don't all come up at once. It may continue over a long period, but eventually, hopefully you'll get good results like you see in the flat of this picture. The little montage of photos at the bottom of this slide shows my results with Adenia Balii. The seeds are quite large. I have a little pot of seedlings there on the right. And after 3 years, they make pretty nicely sized plants like you see in the middle. And after 9 years, the plants are large enough to fill almost a 12-inch pot. So this can be a very satisfying way to grow these plants. The best and most rapid growth is achieved when you give them plenty of root room. They like to have that root space. Give them a well-drained, humusy soil. Fertilize them every time you water them, and maintain a slightly acidic pH in your water when you do water them. So that all sounds very successful, but the frustration part of it comes in when sometimes it just doesn't go that way. You know, I have a picture on the left here of a bucket of Adenia spinosa from a few years ago. At that time, I had close to 100% germination. But this year, my photo on the right shows that I haven't gotten any germination whatsoever. And I don't know why. It's very frustrating. And this is an experience that many people who try to grow these plants from seed experience. And that is just unexplained failures occasionally that nobody has a solution for. So let's move on then to propagation by cuttings. And I have 3 different species in this slide that I'm currently propagating by cuttings in my greenhouse. It's very easy to root adenias from cuttings, as long as you use a thick stem and you take that cut during the growing season. Just dust a little rooting hormone onto the cut, let it callus for a couple days, and put it into a rooting medium. They're not very particular about what rooting medium you use, but it's good to place the pot on a heat mat and put it in a semi-shady location. Give it a little bit of water to encourage the formation of roots, and especially with the leafier plants, it's good to mist them so that you avoid desiccating the plant. One of the benefits of taking cuttings off of adenias is that in many cases, the cutting will be more floriferous than the parent plant that it was taken from. So it's a good way to produce seed. Another reason to take cuttings on adenias is that it's a great way to create a new plant. The plant in the back in this example is an Adenia Patchouli. It was taken from a cutting 14 years ago, a cutting just like the plant in the foreground. So you can see that in a fairly short time, it's developed a very substantial caudex that is exactly what you would hope to see if you had grown one from seed. So very successful in this case. However, it's dependent on which species you use as to how satisfactory the results will be. Here's an example of an Adenia Globosa. On the left, we have a cutting taken from the plant on the right about 10 years ago. And you can see that what has happened here is too much of the growth has gone into the stems and not enough into the caudex. And so it produces a unnatural appearing plant that's not very satisfactory. And even worse than this is, and my example for frustration is Adenia balii. The cuttings from Adenia balii do root very easily, and they're very floriferous, but the problem is that the fruits are so large that if you have a female cutting, there's not enough energy in the cutting to support the fruit. So the fruits abort. And then an additional problem is that the cuttings never expand in size. So they don't develop a caudex, and eventually they die. So this is not a good species to try to grow from a cutting. It's best to grow it from seed. But the catch-22 here is that sometimes it's very difficult to get any seed if you can't get the seed from a cutting. So this is a very difficult species to propagate all around. And that is my rather quick presentation. And I'm going to stop sharing my screen.
Thank you, Rob. I appreciate it. A couple questions for you from the audience. In your experience, approximately how many years from seed does it take Adenia balii to flower?
Well, my experience is that the males are much quicker to flower than the females. Right now, I have 2 males that I'm getting lots and lots of flowers off of, have been for many years. These plants are about 10 years old, I think. They've been probably flowering for at least 4 or 5 years. I have a female plant that has only put on 1 or 2 flowers, and I'm really waiting for it to become more floriferous.
Thank you. Also, what is your preferred medium for germination?
Well, I've tried different things. I've done some experimenting with just straight pumice. I've tried my regular potting soil, and it doesn't seem to make any difference. It seems to work pretty well either way. I think these plants are very easy to do from cuttings.
Got it. We'll come back at the end in the question and answer. Thank you, Rob. We'll keep moving. Our next speaker is someone who has worked at the Huntington for the last 38 years and is currently the curator of the desert collections. Prior to working at the Huntington, he trained with master propagator Frank Horwood at Abbey Garden Nursery in Carpinteria, California. His horticultural writings have appeared in the Cactus and Succulent Journal, the Euphorbia Journal, and various other horticultural publications. He's also known for his photography, with over 3,000 of his images having been published. John also oversees our annual ISI Introductions program, which is a long-standing plant introduction program here, which is now published in the Cactus and Succulent Journal. He holds a bachelor's degree in horticulture from Cal Poly Pomona, and before that studied botany at Santa Barbara City College in UC Santa Barbara. Welcome, John Traeger.
Thank you, Sean. Okay, so today I'm just going to run through a couple of quick notes on propagation experiences from the last year, and hopefully there will be something new in there for some of you. So cactus grafting, of course, has been employed for many years as a way to increase the growth of less vigorous plants by putting them on a vigorous rootstock. But also it's very useful for saving plants that are suffering from one malady or another. In the case of the Schick hybrids, for example, we've been experiencing a lot of fungal problems growing them out in the shade house, where the temperatures are cool much of the year, times of the year, and the humidity is higher. By growing them indoors in a hoop house or greenhouse, we don't have the same problems, but we suffered a significant loss of much of our stock of some of the hybrids, and are doing our best to save these historic cultivars for perpetuity. So Candlelight is one example of a cultivar that's particularly susceptible to these fungal problems, named for the candle-like appearance of the plants in bud, in flower bud. So we did manage to get one clean offset or a few clean offsets to work with for grafting purposes. In this case, grafted on Trichoceres Fieldianus, which is a commonly used stock. And the significant thing I want to point out here is that with a single offset, like this one in the back, you can do a cross section and graft the top portion, as you see in the front and in the back. But with the bottom portion, you can invert it and graft it upside down on top of that stock. And this is a couple of months after grafting, and you'll see it's already squirting out new offsets. So that's a way to maximize a limited amount of scion material. This is another one that we've done a little bit of grafting with, in this case on Clyster cactus Fieldianus, which is a nice vigorously rooted columnar plant. The choice of grafting stock is not too crucial, except it should be something with a vigorous root system, theoretically more vigorous than the scion. And I prefer a short columnar plant with some spines, so that when you put the scion on top of the stock, you can hook the rubber bands to hold it onto the spines wherever the length of the rubber band takes you. So you don't have to worry about wrapping it around the bottom of the pot with a particular size rubber band. So this is a particularly beautiful form of Parodius autunus, and you'll notice in the flower shot on the right that those stamens are rather thin, the anthers, because they lack pollen. This seems to be a male sterile selection, and I haven't gotten any seed using pollen of this plant. And the females are only partially fertile. I've only gotten one batch of seed by crossing it with a typical yellow flowered form of autunus, and we'll see what those seed produce for us. But we're working it up vegetatively in the meantime. There are various specialized pollination requirements for particular kinds of plants, and Oncorina's are one example. We introduced many of these species through ISI thanks to Swiss explorers, Walter Rusli and Ralph Hoffman, who went to Madagascar every year for about 15 years and found a lot of incredible new things, grew them incredibly well in Switzerland, and then we were able to import them. This is one that we never had enough of to distribute through ISI, Oncorina leandri. Fortunately, we have a couple of clones, and outcrossing works best for these, although occasionally self-pollination can work as well. It's a particularly rich yellow color, and it has very distinctive fruits with this wide spineless flange, but the same sort of harpoon-like projections that latch onto the fur of passing lemurs and are distributed that way. So we're gradually working up stock of this one for future introduction. Euphorbia Gymnocalicioides is another one that's not very readily available in cultivation. And I was talking with Tony Yoakum at Arid Lands Plants, who does all of their Euphorbia pollination. And she was wanting to get more plants to have more of both sexes, thinking it was dioecious. But it turns out, looking back at an old photo of our plant that we grow in the conservatory and have for many years, that it looks like it's a male most of the time. But in past years, when I took a photo, lo and behold, it had lots of fruit on it. And I really should have harvested it then, but got distracted with something else. So now that we know that it is dioecious, it's just a matter of timing. The males come before the females, in this case by a couple of months. But thanks to a loan of a plant from Barbara Hall, I've been pollinating and getting a smattering of fruits. And by bagging the fruits, we're hoping to catch the seed. It turns out that the seed is so small and ejected with such force that a lot of it escaped right through the mesh of this particular bag, but we're trying another method this year to harvest the latest batch. We sowed these few seeds we got last year in vitro, just on agar, which worked quite well, but another consequence of the small seed is that there's a minimum of endosperm to sustain the seedling. so they don't last very long on pure agar. So we'll be trying it with more nutrient-rich medium the next time around. Then I'd like to just mention that there are occasions when things set seed serendipitously in the garden or in your greenhouse, which sometimes are worth growing. Often they're open pollinated and who knows what they're hybridized with, but so it depends on the group. Aloes and agaves, for example, are notoriously prone to hybridization, but certain euphorbias are not. Now, this is a plant that is commonly grown, easy to propagate vegetatively, but I'd never noticed it in fruit before, perhaps because I never looked closely enough because it's such a common plant, although we admire it for its leaf color. But I did notice some fruit set this year and decided to bag that and grow it just to see what percentage would come up with the nice coloration of this form. It also might serve nicely as a stock for grafting the Euphorbia Gymnocalicioides in vitro. So we'll grow some of these seeds in vitro. And here's another plant, Euphorbia Nobele, which is not quite as common in cultivation as the former one. But it set a very nice crop of seed out in the garden this year. So one of my gardeners, Faustino, rigged up a very nice tent with a trough at the bottom to collect the seed when they explode. And finally, I'd like to just mention Viscum minimum, which grows obligately on certain species of euphorbias, Euphorbia polygona, and we've used Euphorbia polygona snowflake, the white epidermis one, because it's such a nice contrast to the red berries of the Viscum. And here you can see the stems of the Viscum just emerging out of the epidermis of the Euphorbia. Most of the tissue of the Viscum is inside the Euphorbia, and you don't really notice these small stems much, but the berries are very noticeable. And unfortunately, the birds notice them too and will feed on those. Hopefully, when our new conservatory is renovated, we'll be able to keep the birds out and hold on to our Viscum seed. So that's all I have. Thank you.
Thank you, John. We'll keep moving in light of the time.
That's fine.
Okay. And our last speaker is the 2018 recipient of the Paulecki Junior Commercial Award that recognizes individuals whose commitment to the highest standards of excellence in the field of commercial horticulture contributes to the betterment of gardening practices everywhere. He is the president and general manager of San Marcos Growers, a wholesale nursery in Santa Barbara, California. And over the years, he's helped build a reputation for high quality, sustainably produced plants, particularly those that thrive in California's Mediterranean climate, while inspiring other nurseries to improve their own production techniques. He also promotes climate appropriate plants through his business and several trade organizations to which he belongs. He is a past board member of the CSSA and spoken before at this symposium in 2012. Please welcome Randy Baldwin.
Okay, I want to thank Sean and the Huntington Botanic Garden for asking me to speak today. And when I was first asked by Sean about this, I thought about doing a Perculacaria decarii from root cuttings, but then I found out Joe Stead was speaking as well, and Joe was the one that told me about how to do it that way, so I didn't think that was quite appropriate. But so I went on to the agave Victoria Reginae albomarginata idea. And then I noticed Sean actually put it on the actual symposium announcement, which is great. But that picture itself was a plant that we kind of started out with propagating this plant. And we certainly are not the first to propagate it. And I'm going to talk a lot about how the challenges we've had and doing this plant. It's not so difficult to propagate. It's just a matter of getting enough plants out there to sell. The plant, let's see. Here's a couple plants. In the 2016 symposium, I put this on the auction table. And then there's another one that we've actually put on the Santa Barbara auction table. And that's kind of how these plants came about for most of the collectors. You would get them at the auctions or the plant sales. And I myself, in the 90s was doing this, and I think I paid as much as $100 for a small 2-inch plant. But that's the way this plant kind of came into the trade. And that's why I'll talk a little bit about its propagation and why it remains fairly scarce and still commands a fairly high price. I also wanted to talk a little bit about our stock plants, where we got them, and why we retain the cultivar name Alba marginata for this plant. Those of you who ever knew Alice Wadehofer, she was a fantastic cactus and succulent grower in Stockton, California. And I acquired her collection in 2004. And with it came several of these plants, and one with a very nice label, which is gone now. But you can barely make it out. And those of you who knew Alice, she had a very distinctive tag, and she always had a lot of information on it. She'd have the date she got it, the person she got it from, when she last photographed it, even might be on there, the price she paid. But anyway, this plant was obviously purchased from Bill Baker in October 1999 with the cultivar name Alba marginata. Now, Latinized cultivar names given after 1959 are not legitimate, and we would not use a name such as this for any plant we named ourselves, but for historical purposes and to honor Bill Baker, we've retained this name. I respect that some will note that this name is erroneous, and I can't argue that they are wrong, but I just want to make sure that I explain why we continue to use this name. That very same plant, 2006. Here it is 15 years later, grown considerably. It's a beautiful plant, obviously. Bill Baker himself, shown here in 2007, is talking about one of his famous Dickia hybrids, and photobombed in the back. You might notice Jeff Chemnick and Hans Hansen. We were all there together at that time. And obvious in his collection, and to this was the plant to the left of him, which was his main plant of Agave Victoria Reginae Alba marginata. And I had received several other plants that came through Bill Baker, and he passed away much too soon. We all know. Many of us were at the Tucson CSSA convention when it happened, and there are so many questions to this day that I would like to ask him, and I can't. So anyway, propagating this plant, we do it by division after the plants have been cored, and coring is a technique that scours out the center of the plant, damaging the area where the apical meristem develops. The trick really is to maintain a lot of nursery stock on this plant. We don't get that many shoots out of each one, and so we currently have about 500 1- and 2-gallon stock plants to work with. And here our propagation manager is working with our agave propagator to select a couple to show the coring process. We've used a sharp knife here. He's going to show cutting it straight down into the center of the plant, right down into the middle, coring it out, taking that central leaf out. We've also tried to drill in an apple core, but we had best luck with this type of knife. We use a longer knife with other agaves, and often that promotes multiple new shoots out of the center of the plant. But for this plant, we tend to use this smaller knife, and we actually get side shoot development and a new primordial leaf emerging from the original rosette. So the plant just keeps going. Here the plants on the left are the ones that were cored, the debris in the foreground. 2 plants that would demonstrate what size we actually sell it as. And then again, that one specimen plant on the right. The picture showing the above view, you can see how the center of that plant is scored out, 2 or 3 leaves removed, and the base of the plant actually scoured. About 6 months later, we start to get this development of side shoots out of the plant. And so this is time for us to divide from it. And so you can see these have been excavated out of that pot. And then spread out, and that's that same plant. And you'll note the plant on the right, that's the original cord plant. It's starting to look pretty much like a regular plant with the center leaves coming out. We plant that back up into a 1 gallon container, put it back into our propagation stock. The two below that in the picture on the right are ready to go into the sales area in about 3 or 4 months before they are fully rooted out. We get also a lot of these smaller ones, which we will tend to put into a liner pot like shown there, 2 1/4 inch rose pot. The soil we use is our standard succulent mix, which is a redwood fir mulch mix with washed plaster sand and pumice. And yeah, that's it. So the ones that go into the perlite are interesting. We put those in our propagation greenhouse, which has a little bit more humidity, but we've noticed this kind of interesting phenomena. You can see on the right how the roots actually push the entire plant well out of the perlite. So we need to work on that a little bit differently maybe, but what we're doing right now is just going back regularly and resetting them back in, or at that point putting them into regular soil. The goal of this, of course, is to get sellable plants, and this is about the size of the plants we have for sale. We do it in a 5-inch pot. We finish for our wholesale customers. And since 2012, we've sold a little bit more than 2,500 of these, and they mostly go to retail nursery and online resellers. We've steadily increased our production of it, and we're right now able to produce about 450 to 500 plants a year to meet the demand, or partially meet the demand. And we have a long list of our existing customers that are waiting on these plants to come about. One of the things we do note on it is this chimera. It's a chimera variegation. Almost all these monocots are that way. They show instability and some pure albino sports. And chimeras are, chlorophyll-filled cells adjacent to those lacking chlorophyll. And so as we damage that meristem, we force new shoots, and we often get cell development of just those areas lacking chlorophyll, which produces these albino plants. And while somewhat attractive, there is no way that that's going to survive on its own without the plant that has some of the chlorophyll. And surprisingly, though, we don't get any pure green ones. And that's, I've always thought we should get some pure green ones going back to the regular Victoria Reginae looking plant. This has plagued tissue culture too. In 2007, the Agave Summit was held at Alan Repasey's house, and we got the opportunity to check out Xeric Grower's micropropagation lab. That was a collaboration between Alan and Kelly Griffin. And in that lab, they had initiated this plant, and it looked pretty good. They had a lot of multiplication of plants. But unfortunately, as you'll note on the right, of pure white ones, and on the left, they had a lot of the pure green ones. And I was told at the time, I think I got this right, that they had about 5 to 10% that actually were really variegated plants. And actually, that might have been enough if you could consider how much that this plant generally gets in the marketplace. Looking at the 2016 show, the CSSA show in July, you see that it won the best variegated plant. And I'm really sorry, I can't read the label on that name, but somebody can tell me whose that plant that was, that would be great. My own plant was on display in the Santa Barbara show itself in the same year. And I'm going to end it with a picture of a plant that is in the Huntington garden greenhouse. This was taken during the symposium in 2008. So I imagine that plant is a lot bigger at this point, and I'm hoping that John Traeger can send me a picture of what it looks like. I want to thank you guys for listening to this, and it's been a pleasure being at this meeting.
Thank you, Randy. Fantastic. If I can ask all the presenters to be available, I'm going to get through a couple questions here. I know we're running a little behind, but we'll try to get to some of these. There was a book. Could you please show the book again? Would you please provide the name and author of the cultivation book again for Joe Stead?
Oh, geez.
Or at least tell the name, just say the name of the book and the author.
It's, hold on.
Sure.
It's called Cultivation of Bursera, and it's by Jason Eslamier, Eslamier. M-I-E-H.
Thank you, Joe.
Welcome.
For Brian Kimball, Brian, can you hear us?
Yeah, I can.
Fantastic. How much gypsum are you adding to the soil mix, and what percentage of the mix is comprised of gypsum for Brian?
The basic mix is largely pumice, 80% pumice and 20% regular potting mix with a little bit of horticultural charcoal mixed in. So we just got gypsum powder and sprinkled that on the top, and then we watered it. Then the gypsum would work its way into the mix.
Fantastic, thank you. For Joe Stead, do you have to use a whole tuber when you propagate, or can you use a half, like in a potato, or a little piece of the tuber for Joe?
It depends upon the size of it. I don't like to cut them up too much if they're thinking about, well, I can split it and make two, because you need those roots down below. You need a section of actual roots that come out of the tuber. So if they're thinking about cutting it off and forming two, it's just the case that it has to have some roots attached to it. But I would say you can do fairly small ones. I've done some pretty small ones. You know, the tuber itself may be the size of a quarter or a nickel as far as around, and maybe, you know, 1/4 inch long.
Got it, thank you. For Randy, how deep do you cut out from the center when coring a white rhino?
Well, that is a tricky thing. You got to go down and deep enough to do the damage that you want to have happen. So you've really got to cut out the base of that emerging leaf. But obviously, if you go down into that plate too deep, you're going to cause too much damage. So it's really a kind of a delicate thing. And you know, if you went down in the middle and just took out a fairly deep and left the sides a little more, you would still do the same thing, and you would not kill the plant. So that's what I tend to tell my guys to do, is not get too wide at the base.
Thank you. For Rob Skillen, do extremely xeric species such as Adenia Patchouli prefer cool temperatures to induce germination?
Well, I could answer that question if I had ever had any seed to try germinating, but I haven't. So I'm afraid I can't really help you with that one. Sorry.
That's fine. Joe, back to you. What is the best month for making bursera cuttings?
Well, I think the best month would be in the early spring, when the plants are leafless and maybe just getting ready. You can see them, kind of the buds swelling up a little bit. That would be the best time.
This is to John Traeger. How do you manage plant exchange for international species to add to your collections? Actually, to anyone, but I think it's geared to the Huntington. How do you manage plant exchange for international species to add to your collections and displays?
Well, that's a good question. We're relying primarily on historical collections that have come in the past with permits. Now it's much more difficult to acquire permits, but there are legal means of acquiring new material through botanical garden exchanges and other means.
Thank you. How about this? To all the presenters, I got a question here. What's your favorite succulent? It had to come.
Derstenia gigas here.
Anyone else want to say something? I know it's a tough one.
I probably have more specimens of Aerocarpus retusus than anything else, so I'd have to say it's my favorite. But there's so many things I like. It's I would hate to exclude anything.
Yeah.
Depends on the week.
Yeah.
Well, I would say you can't beat Aloe polyphylla.
Good one.
Oh, I've got one. A seed grown from John Livranos, Adenia Socotrana. Yeah, that's one of my favorites, because I got to grow it from seed, and it's like 10 feet tall now. It's one of my treasures. So it'll probably go to the Huntington.
Awesome. Randy?
Wait, you've locked my video out, so you can't see me, but
Not intentional.
Can hear you.
oh yeah, okay. No, okay, there I go. Well, why not? I'll go with the agave I spoke on. What the heck?
I think that's it. As we're going to pay attention to the time here, I did want to make one announcement. First of all, thank you to all our presenters. Thank you for making the time to show up and do this and get through this symposium. We're really glad that we offered it again this year in this virtual format. So thank you, thank you, thank you. We're just about out of time with our program. I did want to end with a moment of silence for a member of our community who recently passed. Mary Irish died on Wednesday, August 18th in San Antonio, Texas. Some of you may know her for her Agave and Yucca book. She was a close friend to several staff here at the Huntington. She worked at the Desert Botanical Garden in Phoenix for many years. She authored 11 gardening books in her lifetime. She was focused on landscape design. And she also has a memoir of her life and garden in Scottsdale called A Place of Our Own. So we remember Mary, one of our own that recently passed. Thank you again, everyone, for joining us. We're going to sign off now. I appreciate you coming. We'll see you next year.
Thanks, Sean.
Okay.
Thanks, everybody.
Okay.
Bye.